Okay, I think we can get started. So I think the first one, Sucharita, is it a student here? Okay, what about next group? Joe's? Okay, all right. So how are you gonna present, using all computer? Can you join the Zoom? I don't know. Can you join the Zoom? Then you can use your computer to present. Yeah. Do you have the Zoom link? Yeah, it's okay then. Let him, because I already started. Yeah. Okay.
Oh, okay.
I'll give you notice like 1 or 2 minutes.
Perfect. So hi everyone, my name is Jose Gonzalez. I'm a PhD student in computer engineering. And if you haven't seen me before, it's because I'm from the online section. So today I'll be presenting my project. It's called Comparative Analysis of Diffusion Models for Enhancing Alzheimer's Disease Detection. So for the agenda, I'll start with the problem, the related work, the data set, the methodology, the results, and at the end, the future work I can implement. So for the problem, the main issue when training medical imaging machine learning models is the lack of good enough data sets, either because of the privacy of the patient or because we simply lack enough information or data. So, and this is really important when it comes to medical imaging machine learning models, because these need to be really accurate so they don't represent any health concern to the patient. So, and to train any machine learning model, one of the main requirements is, of course, having a large enough data set. This can be solved by using diffusion models. So the diffusion models can offer a solution for this by generating realistic synthetic images that can be used to train medical images, medical machine learning models, right? So for this study, what I did was implement 2 different diffusion models. One, a denoising diffusion probabilistic model, and a more novel one called UCAN diffusion model. I implemented these 2 diffusion models to generate synthetic images and use those synthetic images to train a VGG16 model that detects whether an MRI image has Alzheimer or not. So in terms of related work, for diffusion models on the medical field, what is used more is called UNet architecture, which has been really important in advancing medical images generation and segmentation processes. And for diffusion models, how they usually work is they introduce noise to a certain image, and they train the model to gradually take out that noise, and that way the model can, at the end, generate these synthetic images, right? So these diffusion models are often used to create images with anomalies and then compare them to actual real images to see if they are good enough or not. And so far, they have proven that they have a strong performance in terms of medical imaging tasks. So in terms of the UCAN model that I implemented, I took this from a paper that was released not so long ago. So what this paper does is combine UNet with a novel framework called Kolmogorov-Arnold networks. So the way it works is, the way this network works is basically it takes a model, it breaks it down in fewer blocks, in small blocks, and then interprets them and combine them in a few selected blocks that can be easily interpreted and efficiently produced. By doing this, the UCAN diffusion model was able to improve, outperform state-of-the-art models in accuracy and speed. And it also proved that diffusion model can, in fact, help with medical imaging tasks. So this is like an image showing how it works. So as I said, the UCAN model can be used for segmentation and diffusion. So it basically grabs a segmentation or an MRI images or wherever, and the image goes through these convolution blocks that at first, what they do is reduce the size of the image, and it also has skipped connections to keep the important feature of the image intact. When it goes through the convolution networks, then it goes to convolution blocks, it goes to the CAN blocks, which do what basically I explained before. They grab the information, they break it down in small blocks, and then interpret them and combine them. So they reduce the number of it, and they are more easily interpretable. After that, they go again through the convolution block, and it generates at the end the segmentation or the synthetic images. So for the data set I use, it's a data set from the Alzheimer's Disease Neuroimaging Initiative. At the beginning, this data set has around 1,200 images from all these classes over here. But I chose to use only the Alzheimer's disease class and the mild cognitive impairment class, because these were the ones that had fewer images. So I used this class to train the diffusion models. So for the methodology, after I trained the diffusion models, what I did was create a VGG16 model to identify whether an image has Alzheimer's disease or not. And the first experiment was basically train this model with real images, just real images at the beginning, with a split of 80% for training and 20% of testing. After that, the first experiment consisted in training this model with just the synthetic images generated by the DDPM model and the UCAN model. I generated 150 images for the AD class and 100 images for the LMCI class. And then for experiments 2 and 5, what I did was replace these synthetic images. I replaced 10% of these images with real images in the following experiments. So for example, the second experiment had 90% was trained with 90% synthetic images and 10% real images, and by the experiment 5, it was 50% real images and 50% synthetic images. I did this to really compare and see whether the synthetic images improve or not the performance of the model. So this is an example of the generated images from the model, from the diffusion models. This over here, this first row is using the diffusion model, the DDPM model, and this is what it gave me. And the second row is with the UCAN model, the generated images. So for results,
Sorry, how do you train the DDPM UCAN model for generation? You are using that data set?
yeah, so to train these two models, what I used was the data sets, the AD.
You are using the real data?
The real data, yeah, of course. Yeah. Mm-hmm. I use the 171 images for AD class and the 72 images for, so it was a really small data set. So for the results, this is the performance of the models when it was trained only with real images. And these are the experiments. The first experiment, remember, it was when I trained the model only with synthetic images. The synthetic images I generated with DDPM and UCAN. So as you can see, for the first experiment, for DDPM, it was a little bit lower than the actual baseline. But for UCAN, it was pretty close. As I integrated real images to the training process, the performance got better and better. So, for example, for the DDPM, by experiment 4, which, remember, for this experiment, the model was trained with 60% synthetic images and 40% real images. The results were 96% of precision and an F1 score of 75, which compared to this is pretty good in terms of precision. And for the UCAN model, by the second experiment, it was already outperforming the baseline. And it got to a point of 96% precision and also an F1 score of 87 compared to the baseline. This is an ROC curve that I generated, but that basically exemplifies again the results before. So for the other class, the LMCI class, this one, the baseline, the data set was way smaller for the real images. The real images for this class were only 72. So of course, when I train the model, the precision is very low. When I train the model with only real images, the precision and F1 score is really low. But when I train it with 100 synthetic images for the first experiment, the results are already better than the baseline, as you can see, for both models. And for the DDPM, the best performance it got was at experiment 4, where it got 96% precision and 75% F1 score. And for the UCAN score, UCAN model, it also got 96% precision and an F1 a score of 87.
So I think we don't have much time.
Yeah. That one was the last one.
But I do have one question.
Yeah.
So can you go back to the previous slide with the result? So why here? So the result you have, that's for the entire data set, like precision recall F1, and the table above is just for one particular class.
So this is for one particular class. This is just for AD, for both tables.
Why the next table they have the same result?
No, it's different ones. Or did I confuse him?
Yeah, but here the table.
Oh, yeah. Let me see. Yeah, I confused the table. I have another one, yeah.
You copied the wrong table?
Yeah, I copied the wrong table. Yeah, I have it over here. I can show you the other one.
Okay.
If you want, or no.
Okay, so yeah, I think this is interesting. Seems like the generated data is able to help with the model. But I think one question maybe you can think about is, it seems like not all, like why, because the experiment from 1, 2, 3 to 5, right? So I understand you keep adding the real data, right?
Yeah.
So at certain point, why adding more real data will hurt the performance?
Yeah, I'm not really sure about that. I have to look more into it.
Something you have to look into and make sure the results are accurate.
Yeah, for future work, I was planning to integrate more evaluation metrics to determine that, to see what's happening.
Mm-hmm. But I want to see this reasoning, because I may question you this if you write a report like this.
Okay. Yeah, I look into it.
Okay. All right, thank you.
Okay.
Okay, all right, so let's go to the next one. Maybe you can come here. Are you gonna using your own laptop or? What's your name? Here, right? Yeah. Okay. You're using PDF? Okay. How do we do the slideshow? What is this? Anyone knows? Which one?
Like maximize the screen.
This one?
Yeah.
But still like small, okay. Yeah, maybe you click on this to go to the next one, okay.
Like, can I place the mic here? Or
you can just
place here. Yeah. So hello everyone, I'm Sucharita Vemuri, and this is a research prototype model. So I'm going to present a FEB-MNET dual pathology classification reduction using CT. So at first, what is CPFE, like combined pulmonary fibrosis and FSIMA? So it is a complex lung pathology disease which is having overlapping conditions of both fibrosis and FSIMA. So at first, before going into CPFE, I would like to explain what is fibrosis. So fibrosis is which is a rare and complex lung disease which occurred mostly in older old populations. Here you can see the fibrosis pattern which is occurred here in the lower lobe of right and left lung. Mostly fibrosis occurs due to aging and also due to the oxygen exchange, reduced oxygen exchange. And then about FSIMA, which is a condition occurred due to like prolonged exposure to smoke or chemical. So here in this CT, you can see in this sample you can see the air sac which is prolonged elongated in the like upper lobe. So this both conditions may occur, which is like subject to rare. So my problem statement is like develop a model which classify and predict patterns of this fibrosis and eczema. So CPFE is already a rare condition. There is no data sets or data which we can use. So that's why I used both different fibrosis and eczema, not a combined one. My model will be like classifying and detecting fibrosis and eczema both collectively. And why I choose this, like again the same, underexplored this domain. Mostly everyone will be, most models we can see is like nodal or cancer or COVID-19, which we know all about deduction and biomarker identifier, but not about the fibrosis and eczema, which is like one of the rare conditions. And according to Global Obstructive Pulmonary Initiative, which they stated CPFE is one of the third scale mortality worldwide. And then moving on to the related work, first this harnessing, the first paper is what the idea is inspired. They are the to my knowledge, they are the only one they provided the idea of how to classify and deduct CPFE using deep learning methods. So they used 3D ResNet and 3D Swin transformers to classify and predict. So they use 3D ResNet as a feature extractor, and then those features are collectively used to train 3D Swin transformer to classify and predict. Apart from that, they used quantify the CPFE score, and they used a data set which is like in-house licensed data set, so I can't able to find those data sets. If the licensed data set is available, then we can develop and do more research on how to quantify, predict, and one-to-one learning framework. And then Fibronet, they used 4 different VIT transformers to classify and deduct fibrosis, only one pathology condition. They attain like 94.5% accuracy. And VIT, ILD. ILD is again interstitial lung diseases, which has all the diseases, not only fibrosis and eczema, which includes all nodal and all. The pulmonary is one of the broader area. It covers from respiratory system to heart. So they used ILD dataset, which they classified and deducted eczema, fibrosis, and other lung-related diseases. So they used DenseNet feature extractor, and they used the same methodology, and they achieved 82.75%. And then another one method which used VGG and CNN to deduct lung diseases. And moving forward to end the related work, so the idea is to use a feature extractor, because fibrosis and eczema both have very intricate patterns. So we have to give importance to the features which are present in either 2D CT or 3D CT. So the idea is to have a feature extractor and then a model which can able to learn and distinguish those patterns. Moving on to next is the thing. See, I can't able to find, that's what I said. So I selected 2 different data sets, which is fibrosis data set I collected from Kaggle, which has 3 splits, train, test, and validation on 2D CT images, and eczema data set, which consists of very less samples, but it helped to analyze the patterns of eczema, which has 4 labels: normal, lobar eczema, parasital eczema, and panolobar eczema. These 3 are one of the next rare conditions of eczema within it. So these 2 are collectively used to train and classify the model. And moving on to, before moving on to the model, these are the steps I followed to prepare the data. First is cropping. Cropping is why is cropping is we can generally give a random cropping thing, but I chose the 15, 8, and 12 with manual calculation of the CT images which I got, and then resizing and contrast enhancement only for the FSIMA data because it lacks some features which lacks some the brightness and all those parameters, and color augmentation only for the training purposes, and then all this data are then converted into tensor. Moving on to methodology, this is what I followed. So the 2D CT images collectively added with like resized into 22:4 cross, and then I used a C-SWIN transformer for feature extractor. So this model is only used as a feature extractor because SWIN, we all know, they have good feature extracting properties. So this feature extracted are then concatenated and used for classification purpose. So my backbone classifier is MEDVIT, is again a vision transformer. So the concatenated features are then added to the model using late fusion strategy, where MEDVIT will be initially trained with the total amount of data, but those features before moving into classifier will be used as will be having this features and MEDVIT feature. These both features will be again verified using a data rewriting strategy. So this, what it happens is that 0 and 1, where if the parameter tuning goes close to 0, the feature will be a false. 1, if it goes to 1, it will be a positive feature. And then those features are added to the classifier head of the MEDVIT, and then classification happens, and we get the result. This is the top view of my method. C-SWIN transformer will be act as a feature extractor. So main, like as a time constraint, I will explain the main block. So the main is the attention block, local enhanced positional encoding with cross-shaped attention. As MEDVIT, even MEDVIT will be having the patch embedding and all those layers. Why I used C-SWIN as a feature extractor is that I felt the FSIMA data is having less quality than compared to fibrosis. So I used this model as a feature extractor where as the attention of given to an image will be in like pixel by pixel, patch by patch. Here, this C-SWIN will be going in a cross-sectioned. So the image will be converted into a cross shape, and then each blocks of the image will be giving attention, and those features will be extracted at the end. And then these features are then used to MEDVIT. So MEDVIT coming as a backbone architecture, it has this blocks. First is efficient convolutional block and MHCA, multi-head convolution attention. So here the initial training data will be used again to train all the feature extraction and patch extraction process. Here like more importance will be given which patch to give importance and which patch to be which feature to be reduced. So and at the final layer, the extracted features from the C-SWIN will be like as I explained will be concatenated and using data reweighting parameter. Here you can see in the formula. And I conducted the experiments like taking the old samples from both the fibrosis and FSIMA. As you know, yeah, there will be a data imbalance, but that's why I used an additional feature extractor to extract the features. And with 60-30-10 ratio, the training and validation testing splits are taken. I didn't choose the entire fibrosis from the original data set, but I chose like manually selected fibrosis data and FSIMA data. And evaluation metrics as you have seen classification. So I followed the standard accuracy, precision, recall, and ROC curve to evaluate the model. So this is the result at the initial stage. So this the T-SNE graph shows how well the C-SWIN extracted the features. You can see, so 0 is the normal fibrosis patterns, and 1 is the fibrosis patterns, and 2 and 3, 4 are FSIMA patterns. Sorry, 2 is the normal FSIMA pattern, and 3 and 4 are the other FSIMA condition patterns. And then this is the training validation logs. So training and validation at the mean, both fibrosis and FSIMA really went well. So you can see the loss and accuracy like increased in each and every step. And then of results, individually I evaluated the MED-V8 in the old learning framework in fibrosis data set. This is the results. It performed really well, not so bad. There are some misleading pattern misclassification, but mostly it able to classify the like important features. And then this is the result for. Yep.
So I think one thing maybe you can explain is what exactly, why you are using the architecture like that, like the C-swing and all those things. Like what is specifically you are trying to achieve based on that structure. I mean, you don't have to answer right now because I'm giving you the feedback. In the final report, you want to, explain what's your design choice, right? Why you are doing that?
So my aim is to like predict the CPFE pattern. So I didn't use the original data set. That's why I used that architecture to like give more importance to the feature learning part.
Yeah. So you can explain that clearly, and also maybe show some of the examples, right? So like
Yeah.
some of the difficult classes, they maybe have to, maybe the structure is still not able to capture, right? So it's more
Sorry, in the methodology, in this architecture or the framework or?
No, I mean, I'm asking you, like, why you are designing this architecture? Like, are this the same as some existing work, or which part is your new thing? So you need to, present, or at least in the final report.
The existing one are, like, they all focused on one pathology, not on the both.
Yeah, so that's why the distinction, you need to make that clear.
Yeah,
Okay.
sure.
Okay. So let's move on to the next one. Okay. So who's next one? Any student online?
I think I can go next.
Okay, yeah, let's just keep going. Are you joining the Zoom?
Yeah, I'm already in
Okay,
Zoom.
all right. Can you share the screen?
Yeah.
Okay.
Hello everyone. My name is Fazil Kagdi. I am an online student of this class, but I came to present my presentation today in class. I'm going to be talking about my project, which is lung nodule segmentation and classification using CT scans. So the problem statement is actually pretty simple. It's just that lung cancer is the leading causes of cancer-related deaths. So out of 5 people, 3 people die from lung cancer. So that's why early detection of the lung cancer is possible, and it's also very useful. So that's why, my problem statement includes that. The aim is to actually automate the nodule detection and also to classify whether the nodule detected is malignant or not. Basically, is it cancerous or not? So the related work includes the LITC-ID RI dataset. It's like one of the biggest datasets that is provided by the Cancer Imaging Archive. They have over 1,000 CT scans with annotated like lung nodules. All the annotations are provided by up to 4 radiologists. That means that annotations are not consistent. Some places you only have like 1 or 2 radiologist annotations. Majority of the cases have 4 radiologists. So the annotations include, of course, the nodule location and also the segmentation mask, and also the malignancy rating, which is how cancerous is it, which is like between 1 to 5. So this is like a benchmark data set, so that's why I chose this data set. So another related work segment would be UNIT, which is what I'm using for my model architecture. For the first part, which is the segmentation part, I'm using UNIT. I'm using a 2D UNIT because of familiarity, because we have already used it in other classes as well. We already like are familiar with the architecture, symmetric encoder decoder architecture. Then for the classification, I'm using a ResNet model. I'm using like ResNet 18. I'm using a pre-trained ResNet 18, but it's fine-tuned on the data that I extract later on. So one important credit there that I want to give is to this pre-processing tools that I use to pre-process the data. As I said, the data is all over the place, and there are like several annotations. Sometimes it's missing or whatever. So there are 2 important libraries or GitHub repos that I used in this project. One is PyLIDC. It's a Python library for processing the LIDC annotations, and it's able to basically combine everything, all the annotations and everything together and organize them systematically. I also use the LIDC IDRI pre-processing. It basically this basically uses the PyLIDC library to organize everything cleanly, like in a data folder, converting the 3D slices to 2D slices, 2D like axial slices, so the top view. So this is where this is the libraries that I use to pre-process the data. So we have already talked about the motivation, which is basically just to make sure that the initial screening process of lung cancer becomes easier, and it's also slightly automated. Besides that, I also want to create an end-to-end pipeline which is reproducible and also extendable using more data. So the main contributions are, of course, I already went over it, so I'm just going to skip through. So our approach outline is basically I pre-process the data to get 2D axial slices and the corresponding 2D segmentation mask, as well as the rating for each nodule, whether it's like cancerous or not. And then I use the segmentation masks and the 2D slices to train the 2D unit. After training, I basically extract the patches. By patches, I mean the small parts of the images where the nodule is detected. And I use that and their corresponding labels for each of the nodules to train the classifier. And then at the end of the entire pipeline, I visualize it. So as mentioned for the project, the LIDC IDIR dataset is actually really huge. It has over 1,000 CT scans, 1,000 different patients. And so I used a subset of the data. I have only used like 150 patients. That's because of limited resources, because the actual data set is huge and it won't fit in my device. So that's why I used a subset of that data. It has I've already went over how they are organized and everything. So yeah, pre-processing is very simple actually. I also use the MonAI library for the 2D unit to import the architecture and train it. Over here, the important part is the loss function that I use, because in assignment tree, the problem with for segmentation accuracy is that if there is a class imbalance between the foreground and the background, the model tends to converge towards just predicting the entire image as a background or something like that. And that's actually also applicable over here as well. For assignment tree, I could not rectify it because I couldn't figure it out. But for this case, I actually figured it out, and I use a special weighted loss function which uses the dice loss and also the binary cross entropy loss. But in the binary cross entropy part, I actually calculate the weightage of the background and foreground, and I make sure that the class imbalance doesn't affect the convergence of the model. Along with that, I use the Adam optimizer as well as reduce learning rate on plateau, so that the learning rate is systematically reduced whenever we hit a plateau, and we can get the maximum validation score we can get. Also, the early stopping is based on the dice score, the validation dice score, to be accurate on unseen data. So, and then after the detection, I actually extract the 2D masks that was detected. I crop them, and then I resized them to 224 by 224. And as already mentioned, the annotations already contain whether the nodule is benign or malignant. Basically, is it cancerous or not? So I just take those labels and the masks that I have cropped, and then I train the ResNet classifier. I already mentioned I already use pre-trained ResNet, but I fine-tune on the data to output like two classes, which is whether it's cancerous or not. The patches I have divided into 80% training data and 20% validation data. And again, as with the segmentation model, I again use the Adam optimizer and reducing the learning rate whenever I hit a plateau, along with like early stopping or with the patients of 5, so that like I don't keep on training without improving performance. So this is how the end-to-end visualization looks like. As you can see in the image, this is the on the leftmost side we have the original lung segmentation slice, and then on the right next to it is the original ground truth mask, and right next to that is like the predicted mask from my model. And then after extracting the patches, I also have given the classifier prediction and also the ground truth prediction as well. So now let's talk about some quantitative results. So my dice score when I started training the model, my validation dice score, this is what I'm talking about. So it's on unseen data, started from 0.02 and it peaked at 0.75 almost. And same for my classification accuracy. It started actually really low and it peaked at 90% on unseen data as well. So yeah, that's basically like, what my project is. Right now, the final conclusion is that I've made it for like a small subset of the data, just including 150 patients, but it can be expanded to like the full data set with like more resources and storage space if like available. The entire project is also uploaded on my GitHub with like clear-cut instructions on how to download the data set, how to pre-process it, how to train the classifier, how to visualize everything. Thank you so much.
Okay, all right. So, what exactly the problem you're addressing here? Like, you just apply a simple ResNet or UNet to the classification, right? So, like, anyone can do that.
Yeah, but the thing is, anyone can do it, but no one has done it. So whatever has been already done in the literature, it's either simply for either segmentation or either simply for like classification on the nodules. So I'm clubbing both of these together to make an entire pipeline.
Like, how do you do this? Like, you just have two task heads? Like, what is how do you achieve this? I didn't see that. Do you have a pipeline?
Yeah.
You can show, like, how do you achieve this?
Like the results. So I already
how
like.
do you achieve like classification and segmentation at the same time?
So, okay, so the step is actually pretty simple. I first do the segmentation.
I mean, that's the thing I want to see, right?
Yeah, I first do the segmentation. It's actually in my code as well. You'll see that I first do the segmentation and whatever segmentation mask I extract, then I use them to train the classifier, and then like it classifies. So basically, whenever you input a CT scan, my model will first segment the nodule and then it will classify it. and then it will give you the classification rating. So it's like an entire continuous pipeline. It's not two separate models.
Yeah, but I mean, this is can be easily done. Like, why do you say like the other work doesn't do this? Is it easy to do or it's difficult to do?
It's actually like the data pre-processing is actually one of the very crucial aspects of this project. Also, like I would say that.
I mean, think about like tumor segmentation, right? There's one class can be a normal class, right? When you do the segmentation, you also automatically taking care of classification. So I'm not really getting the problem you are stating here. So yeah, we don't have much time to discuss, but I think that's the biggest concern I have. Like, what exactly? Because all I hear is very standard step. Even I look at here is very standard, and you apply UNet, right? Train it, and do the visualization. I don't really see what is exactly something unique you have. Right? The previous, all these previous, presentations, they have something, unique, right? So like, data generation. And last lecture we have people working on, vision language model to understand or build a data set for, radiology report. And also we have, pre-processing how that will help or affect the performance. So I think you need to be very clear, right? So what is the distinction or the unique thing you are doing? Otherwise, it's just another programming assignment, right? So you are doing the classification. So hopefully you can, be clear on me and maybe also elaborate on that, right? So
yeah, actually, this is like one of the suggested projects that were given in your thing, in your suggestions of the projects. So I just picked this up. That's why. But like the unique part, as I told you before, is the entire pipeline clubbed together.
That's maybe something you have to mention that very clearly in the
Yes.
report, right?
Yes.
So the project idea, I just give you the idea. Not
Yes.
necessarily you have to follow exactly the same thing.
Yes.
But I specifically emphasize you need to have something that is unique you need to address.
Okay.
So I think you have, but you just didn't present that here.
Okay.
So because I look at the pipeline, right? So everything's kind of standard. But I think I'm sure you have something there, but you just have to make that very clear
Okay.
in the report.
Okay. Yeah. Thank you so much.
Okay, all right. So who's the next one? So Jaden, are you presenting next? Okay. So
Yes.
can you share the screen?
Okay. Can you see my screen?
Yeah. Okay. Yeah. Go ahead.
Okay, so my project is early stroke detection using diffusion techniques for image enhancement and UNet segmentation. So stroke is second leading cause of death according to National Center of Biotechnology Information. So this is pretty important for trying to use stroke segmentation to basically identify ischemic stroke at an earlier stage, which will then help the patients as the timing is crucial for strokes. And segmentation tasks, especially with MRI images, often struggle with being noisy or low quality. So the challenge is to improve the segmentation through improving these noisy images, or even trying to improve segmentation with limited data as well, or low quality images. So related work, UNet architectures are often popular for medical image segmentation. Over here to the right, we see that this is the sample UNet architecture. It's often used a lot in medical segmentation. And diffusion models have been applied to generative tasks, like in generative AI. They often are used, and there has been research that has shown that through diffusion models, we are able to generate better images that rather than if we were to just try and generate images without it. But there has not been a lot of research on diffusion models for MRI images, including in the medical field with segmentation. So that is why we are going to try and do that with small data sets and with the MRI stroke images. The motivation behind this is often, as I said before, because there's low quality and noisy images, and some diseases don't have a lot of image examples, and the data sets are small, which diffusion will actually help with, and even a transformer, TransUNet, would help with to further segmentation. As you can see, this is part of the data that I had with a 2D slice, and this is kind of an example of how the image could be a little bit lower quality than if we were to not use a MRI image. So the main contributions of this project is integrating diffusion model techniques, such as adding noise for image enhancement on stroke MRI images. And we compare the UNet without diffusion techniques with UNet with diffusion techniques, and we evaluate if these enhanced images will actually improve segmentation performance. The work basically looks into segmentation performance on small data sets through image enhancements. So the approach will be first to process the MRI images in 2D slices and store them, and then use a pre-trained UNet model to extract these features. Then use diffusion model techniques such as adding noise to transform the training data and use that on implement that into a UNet model and compare the UNet model with diffusion versus the one without. So for the data set of this experiment, we're using AL's 2017 data set, which is ischemic stroke lesion segmentation, which is a part of one of the most popular strokes, and the data set is all images of that. It has the patients that have experienced this stroke with, as you can see here on the right side, there's the mask compared to the training images. And often in ischemic strokes, the blood flow to part of the brain is blocked off through a clot, which is leading to brain cell death in minutes. So timing is often crucial. And it could also be important to identify if there is possibility of a stroke at an earlier stage. So segmentation with enhanced images will really help with that. For the data set that I used part of IELTS 2017 off of Kaggle because the full IELTS 2017 data set could not be accessed unless you request access and are accepted. So I used a part of it from Kaggle. This is another example of part of my data set. As you see, subject 30, slice 10. For one of the MRI images, there's a 2D slice, and then there's also the subjects, which is at different levels of that slice as well. So this is at 30, and then there's the corresponding mask for that. So details of the approach, these are basically some of the diffusion enhancement technique that I use, which is adding random noise in the image. And this will try to simulate how diffusion models, and they add random noise and then they reverse it. So in my approach, I mainly just added the random noise to see if that could improve the UNet's performance on stroke segmentation. The baseline that I used was the UNet without diffusion, and this is kind of the architecture or the setup of that UNet model without diffusion. And it's from Monai that I'm going to compare against as well. So as far as the data set goes, there was the data set of the IELTS 2017 portion that I got from Kaggle had 40 MRI images and 19 slices per image. So it's not very large data set, and they were all in grayscale, but they're converted to 3-channel for UNet. And I also used combined laws with DICE and binary cross entropy. And to analyze performance, I also looked at DICE score, IOU, intersection over union, accuracy, and F1 score. I also used, for setting up the training, I used 15 epochs. I used Atom Optimizer and 1E -4 learning rate, along with the loss function, as I said before, combining. And I trained on the Kaggle GPU P100 to make it faster. So for results, when we're comparing diffusion techniques versus without diffusion techniques, we can see that the accuracy is a little bit higher with diffusion accuracy, but it tends to be close to the same. But as you can see in the loss, there's actually a large drop-off between with diffusion techniques versus without diffusion. So it seems to suggest that with diffusion techniques, we actually are able to lower the loss and improve the model, improve segmentation, I mean. So for evaluation as well, we can see that although the overall results aren't great because of the smaller data set, UNet with diffusion techniques tend to do a lot better than the UNet without diffusion. And as I was evaluating over several iterations, you could see that UNet with diffusion techniques tends to be a lot more stable and increasing accuracy and decreasing loss than UNet without diffusion, as UNet without diffusion tended to jump higher and lower in the scoring. So limitations, overall, the smaller data set tended to have limitations on the overall performance of the model, and the loss is both high due to that. And sometimes the data was a bit unorganized, so it took a lot of organization and in setting up the training and the mask. Future work that I want to do for this project would be to investigate into multiple advanced diffusion models, or adding in a DDPM, which will have higher performance, and it's a full diffusion model, so it won't just add noise, but it will also reverse the noise as well. And this will help to enhance the images even further. And I also want to get a larger data set from the OWL site, so get the full data set so I have more data to train on as well, and implement a transformer trans-UNet model that will basically try and look further to segment better as well, since the MRI images are 3D and they're larger and harder to spot ischemic regions. In conclusion, we can say that UNet with diffusion techniques tends to improve the segmentation performance by enhancing the images before passing them into the model. And this could be an interesting thing to look into when we're looking at stroke MRI images and trying to segment and find those ischemic regions at an earlier stage, and could be applied to other MRI images in the medical field. Thank you.
Okay, thank you. Yeah, I have a few questions. Like, I don't really understand, like, what is the method you hear? Like, can you go back to the approach? Like, you have the original data, right? And you have the segmentation mask. What are you trying to do here?
So I'm adding noise to the image. Like, I'm basically trying to simulate what a diffusion model would do. But in the forward step, I add noise to the image, and that basically tries to enhance the image quality so that we could.
How do you can tell? Like, I don't understand. How can you say the diffusion model can enhance the image?
Well, through the process, it's able to.
Yeah, like what is the evidence tells you it can enhance? How do you train the diffusion model, by the way?
I'm not training the diffusion model. I'm basically enhancing the images.
Like, tell me what
So
diffusion model you are using. Are you using a pre-trained diffusion model?
No.
Okay, so how do you use the diffusion model in this case?
So it's not a full diffusion model. It's more of like the diffusion model process. So when you add noise and then you reverse noise, I'm basically doing the add noise step to see if it can improve my overall segmentation results.
But you need to train the diffusion model, right? Otherwise, how do you expect to denoise?
Well, I don't denoise. I was seeing if the first step.
You just said you're using the added noise and denoise it. That's the diffusion process.
Mm-hmm. Yeah,
but
I don't do the full diffusion process. I only do.
Tell me
part.
what exactly you are doing here.
I'm doing part of the diffusion process, so I'm adding noise and seeing if that improves my results.
How can adding the noise will improve the result? You have a clean image, which is original raw data. You are adding noise to the image and hoping that will help the segmentation performance. I couldn't get that. Anyone understand this?
Data augmentation. You add noise and then it's more robust potentially.
But he said using the diffusion.
But like just the adding the noise part, which is just kind of data augmentation.
So you are just adding the noise? That's it?
Yes.
Adding the noise, then do what? Then train the model?
Then, yeah, inserting that into the model and training the model.
Yeah, then that's basically you're doing nothing, right? You're just adding the noise, just very simple data augmentation. And I also, can you go back to the result? Like, I don't see why the dice score is high, your accuracy is pretty low, like less than 1%. Right? The mean accuracy, like 1%. And the dice score is close to 1. Like, how would that happen?
I'm not sure, but I just saw the overall results of with diffusion techniques.
And also,
was better.
like, without, yeah, I think this doesn't make sense to me. Anyone have any comments here? Do you see, is this normal? Do you guys think?
Maybe the dice score is the dice loss?
The dice score is pretty high, right? Close to 1.
Yeah, but maybe the log is wrong. The score should be lost because dice loss is usually 1 minus the dice score. And then it's like.
But I don't think so because here, like, the mean accuracy is 40%. I'm not sure if it's percentage or 0.4%. So I don't believe this result. I think these results are wrong because you cannot get a very high dice score with less than like 2% of accuracy. So yeah, I think you are not, and you are kind of, this is based on what you just clarified, you are not doing any diffusion. Right, you're just adding the noise.
Yeah, it's a diffusion process.
It's not a diffusion process because you said you are not doing the denoising. You are just adding the noise.
Yeah.
Is that true?
Yes.
Yeah, then you are not using the diffusion. Right, because the diffusion process is adding the noise, you have to denoise it. But you're just adding the noise as like data augmentation. So I think you need to make this concept clear, right? So we discussed this in class, and you cannot mix all these concepts together. So otherwise, basically, from what my understanding is that you are just adding the noise to the data, some sort of like a data augmentation, and the result doesn't support that. At least, I mean. The mean accuracy, the dice score seems a little bit, yeah, this is like a conflict result, right? So like with diffusion, you have, without diffusion, the dice score is higher, but the mean accuracy, this is significantly lower. So I don't know, like, yeah, I don't think this is the accurate result. Yeah, I think you need to carefully check the result. And also, when you're writing the report, you cannot do a lot of false claim, right? Because, in the presentation, I don't get like what exactly you are doing. And, after the clarification, it seems like you are not doing a diffusion. So you need to be very clear about what you are doing and also examine the result very carefully, okay? So I think that's my suggestion for you. So otherwise, if you do this in the report, then it's totally failed, right? Because there's a lot of false claim, and the result doesn't make sense. Okay, so that's my suggestion for you. So be very clear about what exactly you do, and you should also check the experiment result, right? So otherwise, because I'm gonna ask the TA to run the code. If it doesn't reproduce, then we have to make sure you are able to get the result. Otherwise, just report what you have and maybe analyze, right? Why are you getting this result? Okay. Okay. All right. Thank you. Let's move on to the next one. Do we, Jeffrey, team? Okay. Are you download the? Okay. Okay, you can join the Zoom.
Hello. Hi everyone, my name is Jeffrey, and we are presenting protein detection via cryo-ED. I'm working with James. Okay. Sorry. The problem basically we want to detect protein structure in cryo-ED images, right? But why we care about detecting proteins? Basically, proteins are like the building blocks of every living organism. But the thing is, like, if we want to analyze them, they're pretty small, like a tenth of nanometers. And also, like, this is a core problem in science. Like, we want to study, like, what's the protein function in, like, medicine, in the body, and so on. And usually, this is performed, like, in isolation. But it's important to understand the interaction with other proteins and organisms in our body. And that's why we want to study it in the native biological context. Previous methods like cryo-EM was able to identify high-resolution structure at the atomic level of this protein, but those are usually done in vitro, which basically is isolation. And this gives you a lot of knowledge for biology, but now in order to do another step in the science, we might be to understand the interaction between that. Cryo-ET allows to high-resolution structure, but also in situ, what basically is with the actual sample with the biology organism. I'm not from biology expert, but in situ means like we have like the actual context. And basically what this is a big thing on science and medicine is that they basically can have study the protein with the actual context. Now, I will explain a little bit more about how this is tracked. Basically, you have the sample. Let me just get the. Sorry. Basically, you have the sample here. You froze them, and then you have the tomography. Then you get the image, and then you have some protein here. You can see that these were the proteins that we have, but not all of them are the same protein. In our problem, we want to classify this protein, and I will show you another example of the detection. This is an example. This is like the X and Y plane. This is the X and X plane, and this is the other one plane. And basically, what you can see here is these blobs are like annotation of these specific proteins. And then the thing is, like, even this volume contains other organisms. I think this is a cell or another protein, but those are not annotated. And basically, this makes the problem pretty hard because first, like, it's proteins between each other, they look pretty similar, but also you have things that you should not classify that. In order to classify that, you need to understand the actual local context of that. And this probably is something that we not easily relate before starting our problem, but I will go, maybe we can talk about that later. This is another example. This is like a denoise image. They have, just to characterize the problem, they have low signal to noise ratio. They have multi-class. There's a dense multi-class annotation. Basically, you have multiple classes, and these blobs are pretty similar. You need to distinguish between these are different, and volumes are full of these structures. Like you have these things here. You can see it also in this thing. They have this protein or structure here that should not classify or is not annotated. Okay. The Chan Zuckerberg Imaging Institute released this challenge like 2 months ago called Cryo ET Object Identification, where they have 7 tomographs with 6 classes. They only score on 5 because the 6 they say that is impossible to detect. And then this is one of the original detection. You can see that they have pretty low signal to noise ratio. Like you can almost see nothing here. There's like some visualization here, but this is pretty noisy. Locally, we use the noise version, and then the Kaggle competition like finished like 1 month ago, and we evaluate the best solution. 10 of 10, 9 of 10 solution they use a unit, 3D unit. And they were using DAT picker, and they use a weak supervised approach on that one. The motivation for our project is we have a first manual annotation of this protein is pretty hard. Even like for they use these ET tomograms to reconstruct the protein, the 3 protein. But annotating 1 is not enough. They need to annotate a lot of them, and then manual annotation requires an expert, and it's very costly because you need to annotate a scale of 1,000 protein, the same protein, in order to have a good 3D reconstruction. Automatic detection of this, we accelerate discoveries, and ideally we want a general solution that can detect novel proteins, and then this can enable a fast reconstruction of 3D. Our contribution is we want to improve detection accuracy, leveraging 2D to 3D adapter for better spatial features using pre-trained models. In order to do that, we changed the architecture to transformer, but this might be a mistake of our problem because transformers are not suitable for this stack. You will see that in our experiment. We think that applying transformer in segmentation for this task was an easy task, and then we take something novel like fast accuracy. We're thinking something like token pruning and other kind of modification. And at the end, we wanted to leverage other data sets and to help to improve our model. Okay, I will leave you to James.
So our base approach is a unit backbone in order to predict segmentation masks. So the overall architecture is we have some input volume or the tomogram. We have particle coordinates for where the center of these different proteins are. And we then create volumetric masks. So these are weak labels because we don't actually have the pixel-wise segmentation per particle. So then we randomly sample volumes within this large tomogram and feed these to the unit, and we train it to match the weak labels from the particle coordinates. So this is the base approach. Our core changes revolve around changing this unit to transformer, as well as exploring different pre-training approaches. So the key challenges, or the key changes that we're making is changing the backbone to a transformer, because if we can change it to a transformer, there's a lot of recent research on improving 2D performance using adapters to 3D. There's also easy weight pruning techniques that we can explore. And then a lot of the foundation models use transformers. And so if we can leverage those for the pre-training data, that could help our method be more generalizable. So our experiment setup, we use a ResNet. Region of interest is only 96. So the total volume is on the scale of 6,000. And so these regions of interest are pretty small. Standard training, 10 epochs, around 1,000 samples per epoch. This means it takes about 5 hours to do full training for the baseline. For the loss, we're using cross-entropy over the masks per label. So these are a volumetric cross-entropy loss. And all classes are weighted equally, despite the scoring being different per class. And then augmentations are pretty straightforward, just spatial cropping, flipping each individual dimension, and then random rotations. So the metric that we use is, or that the Kaggle competition uses that we'll report is F beta, which is essentially F1, but with this new beta squared term. So beta squared, or beta is 4, and in essence, when beta is greater than 1, it biases the F1 score towards recall. If beta was 1, this would just be F1 score. So the metric we're using is F beta, and we consider the detection successful if the detected coordinate is within half of the particle's radius. So for some of the particles, the radius is like 10, and so it would need to be within 5 units to be considered success. The classes in the data set are weighted by difficulty, so there's 3 easy classes with a weight of 1, 2 difficult classes with a weight of 2. So our preliminary results, the first result is on the baseline method that we're replicating. As soon as we try to convert this into like a 2D transformer architecture, we aren't really able to get successful training. And we note that like the transformer training is unstable. So the provided training recipe for this unit has like a lot of details and a lot of like fine-tuning things to get the best score for the Kaggle competition. But a lot of these don't transfer into the transformer training. And even when we look at like using a 3D VIT network with the unit, this training also collapses, but it does at least train some. We also know that the transformers appear to like overfit early, potentially because the data set is pretty small. So this would lead us to like, if we can leverage pre-trained weights, this could help us, because if we fine-tune fewer weights, that could improve. So we do some initial experiments on using a pre-trained weights. So if we just use the existing architecture with ImageNet pre-training, it doesn't work very well. And this could be because, again, the training recipe isn't very good in the pre-trained setting. When we convert over to using kind of like the setup from assignment 3, where we use the segment anything model with a tiny VIT using like LoRa fine-tuning, we do get some improvement, but the code that we ported over is very inefficient. So this is taking like 10 hours to train half an epoch. So we're still working on like improving the efficiencies and actually applying these pre-trained weights to the method. So in conclusion, the Kaggle solutions are very optimized for this data set. And we potentially require more data to like fully train the transformer. And right now, the pre-trained weight initialization doesn't work well. But it could be that if we explore additional data sets, that the Kaggle competition solutions performance falls off. So by focusing on that metric, that can be an easier way to improve on these methods. We have a next step slide, but we'll skip these.
Yeah, so I think this is a very interesting, unique. So you guys found this on the Kaggle competition, right?
Yeah, I think it was one of the recommended.
Yeah, so like the current top performance is using the simple unit.
Ensembled. So I think there's like
how many they have?
Six
6?
models.
Yeah.
They have to resonate 34 to resonate with dynamic.
So can you go back to this?
Yeah. It's not there, but we just using one of them in our tables. But
The results you have.
Oh, yeah. So this result slide
Yeah,
is,
so do you also, so this ResNet,
this slide has better results.
okay, the star one is their ensemble, right?
Yeah, this is on the test split. All of the other results are on the validation
So
set.
if you're only using a single ResNet,
We get like
so
71%.
what's the difference between the second and third?
Second and third is the training time. So the third, we only trained 1 epoch.
I see.
The first, we trained 5.
So in that case, ensemble does make a difference, right? It's
Yeah.
able to help a lot. But now the question is, you guys, when you switch to Vision Transformer, you guys are struggling with the training. Is because of the data? Or?
I think a combination of the data and the recipe. So like right now, we're trying to convert the ResNet like code that we have into transformer training. But more probably, we should be converting a transformer training into this setting.
Yeah, so why is the SAM model doesn't work so well? Because
I think purely it's half an epoch. So like this, the training is like it's taking like 2.5 minutes per iteration, and there's thousands of iterations, whereas this should take like 5 iterations per second. And I think this is due to like how we're porting the problem into like a 2D vision problem.
so here the tiny VIT SAM is is it a 3D model or 2D model?
2D.
So is that, you see the disc, or what about the ResNet? Is that 2D or 3D?
Those are 3D.
3D. So do you think the performance gap also due to the 3D model versus the 2D model?
It could be, yeah.
Yeah.
I mean, we also explored initially using like 3D VIT with the UNet architecture. And that does better. But it looks like the training regime for that isn't quite right.
Yeah, so yeah, I think all these competitions, all these solutions, they are highly optimized, right? So because it's not about innovation, rather how you can engineering to training recipe can make all really work for this data. So yeah, I think you guys did a lot of experiment, with all these different models, right? You also tried this, 2D, 3D adapter model. But I think going forward, maybe you need to figure out. So have you guys decided which one you're going to use eventually? Like, are you stick with this UNetter